All right. Thanks, everyone. I also want to just thank everyone from our virtual space and for our virtual participants for providing questions through the Slido system. I know that we've not been able to get to all of your questions, but just as a reminder that those are all being captured and will be shared with our FDA colleagues for providing answers as well. Just to know, we are trying to embed your questions into the discussion that's ongoing in the room as well. So thank you all for doing that. So please keep them coming as well. So I'm happy to be moderating the session 4 today, in which our colleagues from the FDA are going to be unveiling the highly anticipated, I know you're right, we need like a drum roll thing, to talk about the pregnancy safety study framework. So in this session that's titled The Design of Pregnancy Safety Study, whoops, let me hit that slide, there we go. Design of the Pregnancy Safety Study Framework. We're going to hear again from Wei-Ho Hua, who'll be presenting the proposed framework, and learn also about some of the important study characteristics and factors that are essential to think about and be including in the framework. Following from Wei's presentation, we're going to hear from additional individuals from the FDA who have contributed to the development of the framework that she'll be presenting. So you'll hear from Clara Kim, who's a supervisory mathematical statistician in the Division of Biometrics 7, Office of Biostats in CDER. Leyla Sahin, again, who you've heard from several times today, but who is an OBGYN and Deputy Director for the Safety in the Division of Pediatrics and Maternal Health at the Office of New Drugs in CDER. And Sara Eggers is Director for Decision Support and Analysis within the FDA Center, for CDER as well. So I'm going to turn the presentation over to Wei.
All right, so thank you very much, and thank you for the discussion in session 3. So in session 3, we discussed the individual factors that may be considered to inform the selection of studies. So in this session, session 4, we will share FDA's current thinking on how to integrate different factors in a pregnancy safety study framework. Approximately 5.5 million pregnancies occur each year in the US. Half of pregnant individuals use at least one drug or biological product to treat medical conditions. So typically, at the time of approval, there are limited or no human data on the safety of product used during pregnancy. As a result, for most products, human pregnancy safety data are collected post-approval. So the purpose of the framework is to develop a consistent and a transparent approach to help decide when and what post-approval pregnancy safety studies might optimally be used to obtain timely evidence of safety for regulatory decision-making. Under PDUFA 7 commitment, the framework focuses on non-interventional or observational studies, and this is in parallel with other safety surveillance approaches, for example, routine pharmacovigilance. Combined, all sources of safety data may inform product labeling, benefit risk assessment, and the clinical practice. As a reminder, different types of non-interventional studies can be considered for post-approval pregnancy safety, including pregnancy registry studies, healthcare database studies with or without pre-specified outcomes, and the descriptive studies. The different types of studies and the studies of the same type may differ by data collection, data source, and the study design and analysis methods. So what a given study can do, and if one study is better than another, should be determined on a case-by-case basis. So this is another reminder regarding the difference between signal detection and evaluation. Signal detection is for hypothesis generation to identify a risk. So more uncertainty may be accepted. Signal evaluation is for hypothesis testing to confirm a risk or to quantify risk. With that, higher level of certainty is needed, so the study should have stronger internal validity. As discussed in session 3, 4 factors may be used to inform selection of study. We will use these factors to construct the framework. The framework is outcome agnostic. For a particular drug, the outcome of interest is determined by available information and the regulatory question. This slide shows the preliminary framework to help determine the viable and optimal non-interventional pregnancy studies to meet regulatory needs for post-approval safety data. Starting from available information, there are 2 pathways. In the upper pathway, the regulatory question is defined based on what we know or what we don't know about a potential risk. For a particular outcome of interest, depending on the data gap, the study goal can be signal detection or evaluation in a desired time frame. To achieve the study goal, a study's technical capability is a critical consideration concerning the direction and extent of potential bias. Potential bias impacts both the study's internal validity and the sample size required to detect a statistically significant association. In the lower pathway, based on what we know about the patient population, the product, and the utilization of similar products on the market, we might be able to estimate the expected utilization of this product by pregnant individuals in US over time. Because this estimated magnitude of exposure reflects the overall pregnancy exposure in the US, we need another estimate, that is, of the total pregnancy exposures, how many can be captured by a given study. 3 arrows pointed to viable studies in this framework. A viable study is a study that can meet the study goal with necessary level of evidence within a desired time frame. To determine the viability of a study, internal validity, minimum sample size, and the expected exposure should be considered. Subsequently, among the viable study options, an optimal study is selected, considering timeliness, resource requirement, patient burden, and other factors. The optimal study can be one study or a combination of studies combining different strengths. To take a close look of the framework, we divide it into 5 steps. Step 1 is to determine the outcome of interest in the study goal based on available information. All right. As discussed in session 3, pregnancy safety involves a broad list of maternal, fetal, and infant outcomes with varying relevance to toxicity of a particular drug. Based on what we know or what we don't know, outcome of interest can be pre-specified or non-pre-specified. At the time of approval, available information is typically from animal data and the clinical trials. If a product has been on the market for some time, available information can also come from routine pharmacovigilance in the medical literature. If there are similar drugs on the market, risks observed for those drugs may also inform the potential risk for this drug. The data gap, for example, if it's a known risk, or if there is only some suspicion of the risk, or if we are concerned about an unknown or unexpected risk, determines whether the study is for signal detection or evaluation. Step 2 is to assess a study's technical capabilities. In epidemiological studies, internal validity is always a priority when evaluating the suitability of a study. Suboptimal study design, data, and methods can introduce bias. Since there's no perfect study, it is important to understand what a potential bias a study may have, what is the impact of bias, and if the bias may change the interpretation of results. For a given study, the necessary level of evidence depends on what a study aims to achieve. For example, to identify a risk or to quantify risk. So how much uncertainty can be accepted should be determined on a case-by-case basis. In addition to internal validity, the direction and the extent of bias also affects the minimum sample size. This slide shows the key parameters that we need to specify to estimate minimum sample size. The assumed true risk is specific to the product outcome pair. The prevalence is specific to the outcome of interest. Power is the probability to detect an effect when there is a true effect. Exposed versus unexposed ratio can be 1:1, 1:2, 2:1, or other ratios, depending on how many exposed and unexposed pregnancies we have in the defined study population. Type 1 error is the probability of false positive when there's no true effect. So by allowing a higher type 1 error, it is more likely to have false signals, but less likely to miss a true signal. In the simulation, we also adjusted bias parameters to illustrate how the risk estimate may deviate from the truth and how the minimum sample size may vary in different scenarios of bias. In this example, we assume a true relative risk of 2, 3% prevalence for the outcome of interest, 80% of power, and a 1:1 ratio of exposed to unexposed. For illustration, we use outcome misclassification as an example of bias, which is reflected by the imperfect sensitivity and the specificity of outcome ascertainment. Each row represents a unique scenario in terms of the direction and extent of bias. When there is no bias, the projected relative risk is a true risk in simulation. In the presence of bias, the risk estimate deviates from the truth in different directions to varying extents, depending on the interplay of bias parameters. The minimum sample size also changes accordingly. To highlight a few scenarios, in the top 3 rows, the risk estimate is biased to less than 1, meaning the risk effect can no longer be observed. In these scenarios, the signal will be missed even if there is a large sample size. The bottom row illustrates another scenario where a twofold true risk is overestimated to 3.5. This inaccuracy still allows a signal to be detected, but is concerning for signal evaluation if the bias is not accounted in study design analysis. In general, more bias is tolerable for signal detection, and a more rigorous study is required for signal evaluation. However, as illustrated in this example, in the presence of bias, minimum sample size to detect a signal may be larger or smaller than the true sample size. More importantly, even if there is a large enough sample size, the signal may still be missed in certain scenarios. For example, where the direction of association is biased to less than 1. Therefore, study internal validity is still important even for signal detection. Step 3 is to assess the magnitude of drug exposure and the study's capture of exposure. Magnitude of drug exposure is another important factor. Ideally, we would like to have a precise estimate of pregnancy exposures. In practice, this may not be possible. So instead of precise numbers, we use hypothetical categories to illustrate the idea. In this example, we have 5 categories, from category 1, very rare, with less than 10 pregnancy exposures per year, to category 5, very common, with more than 10,000 each year. The threshold for each category is hypothetical. When the magnitude of drug exposure is very rare, descriptive study may be the only option in this framework for signal detection, and the signal evaluation is likely not possible. When the exposure is very common, there may be many possible study options for signal detection and evaluation. If the magnitude of drug exposure is somewhere in between, registry and database studies are likely possible options for signal detection and/or evaluation, depending on other factors such as outcome of interest, studies' technical capabilities, and a given study's capture of exposure. As briefly discussed in session 3, estimating magnitude of drug exposure integrates multiple patient and treatment factors. The light blue circle represents the target patient population. The size of this population depends on the prevalence of disease or condition. The characteristics of patient population, in particular age, sex, and whether the condition is common in pregnancy, inform how many of these patients may become pregnant. In the meantime, the product and the treatment factors inform how many of these patients may use this drug. Considerations include potential market share of the product and the utilization of similar drugs on the market, if the drug is indicated for a pregnancy-related condition. The toxicity risk, for example, if there's pregnancy-related warning or contraindication labeling, or risk evaluation and mitigation strategy, or RAMS. We also want to highlight the individual treatment decision-making, which considers benefits to patient during pregnancy, what is known about drug toxicity, and the other available options. Potential for inadvertent exposure in pregnancy also contributes to the total number of pregnancy exposures. Another factor should be considered is how long the product has been on the market. So this is an example of a hypothetical scenario to estimate the magnitude of drug exposure. In this scenario, we have a newly approved drug for a serious condition, and this product is the 5th in class. Under the patient factors, there are 800,000 patients in the US. The condition predominantly affects females, with the age of onset commonly in 20s and 30s, and that the condition can flare during pregnancy. Under the treatment factors, there is no pregnancy-related warning or contraindication labeling, or RAMS. Although a relatively small market share is expected given it's a 5th in class, this drug is likely to be prescribed among pregnant individuals, considering the need for treatment, drug benefit, and the lack of evidence of teratogenicity. Integrating all these considerations, it may be enough to assume the exposure will be common in category 4, with approximately 1,000 to 10,000 pregnancy exposures per year in the US. The magnitude of exposure is estimated by patient and treatment factors reflects the total pregnancy exposures in the US. A study's capture of exposure can be thought of as a fraction of the magnitude of drug exposure. This fraction primarily depends on how patients are enrolled or accrued in a potential study. For example, for a pregnancy registry study, it relates to the scope of registry, the recruitment and the retention strategies, patients' willingness to participate, and other factors. For a study using EHR or claims data, it concerns the size of the data source and its relevance to pregnancy. Time factors also influence the fraction that a study can capture, especially in the first few years of approval. For example, it may take time to to establish a registry to start patient recruitment. For database studies, potential data lag should always be considered. This is a simple illustration of calculation integrating the magnitude of drug exposure in the study's capture of exposure. See, the overall magnitude of drug exposure is estimated in category 4, that is between 1,000 and 10,000 per year. For a nationwide pregnancy registry study with successful recruitment and retention, the study capture is estimated at 10%, that is between 100 and 1,000 per year. Similarly, for a large-scale EHR and claims data source covering 40% of the US population, it is reasonable to estimate the study capture at 40%. So using this data source, the study may be able to capture 400 to 4,000 pregnancy exposures per year. Step 4 is to determine study viability concerning if a given study can meet the study goal with the necessary level of evidence within a desired time frame. The evaluation will be informed by a given study's internal validity, minimum sample size, and the expected exposure. So this is a hypothetical lookup table of projected risk estimates and the possible sample sizes based on various parameters. We will use the numbers from this table in the following example. This is a hypothetical example to illustrate the idea of integrating internal validity, sample size, and expected exposure to determine study viability. In this example, the study goal is to detect a signal for outcome X. Outcome X is a pre-specified outcome based on animal data. There are 5 studies in consideration. The first 2 studies are pregnancy registry studies. The difference is that study 1 uses a perfectly designed case report form, while the case report form in study 2 is designed with flaws. Studies 3 to 5 use the same healthcare database, but different methods. In study 3, outcome events are adjudicated by chart review. In study 4, potential cases are identified by a claims-based algorithm without chart confirmation. Study 5 uses a method for non-pre-specified outcomes based on a hierarchical tree of ICD-10 codes. The viability of study considers the likelihood of detecting a signal for outcome X within 10 years of approval. For simplicity, we assume non-differential misclassification is the only bias in these studies. Given different methods, the sensitivity and the specificity of outcome ascertainment vary by study, introducing bias in different directions and to different extents. As a result, the projected risk estimate varies by study. In this example, although the risk estimates deviate from the truth, they are all above 1. So the risk effect may still be observed for signal detection. Due to different direction and extent of bias, the minimum sample size also varies by study. Whether the sample size requirement can be achieved depends on the study's capture of exposure. Study 1 is a nationwide registry study with well-planned recruitment and retention strategies. It expects to capture 10% of the total pregnancy exposures in the US. In this example, the annual pregnancy exposure in the US is estimated to be between 1,000 and 10,000. So study 1 is expected to capture 100 to 1,000 per year. Compared to study 1, study 2 is a smaller registry study, and it may only capture 1% of the total exposure. Studies 3, 4, 5 use the same data source that covers 40% of the US population, So their estimated capture of exposure is 40%. That is between 400 and 4,000 per year. The expected 10-year exposure accounts for slow market penetration in the first few years of approval, the time needed to establish a registry study, and the potential data lag. The likelihood of detecting a signal for outcome X within 10 years of approval considers the study's internal validity, minimum sample size, and expected exposure. In this example, the potential bias does not change the interpretation of results for signal detection, so it's acceptable for all these studies based on the study goal. However, sample size is more likely to be achieved in studies 1, 3, and 5, even if the exposure is captured toward the lower end of the range. So these 3 studies are likely to be viable studies. For study 2, the 10-year expected exposure is between 70 and 700, which is way below the required sample size. So study 2 is not viable. For study 4, sample size may or may not be achieved, depending on where in the range the exposed pregnancy can be captured. So study 4 is possibly not viable, especially if a 5% type 1 error is used for signal detection. So this is a hypothetical example, and it's only for simple illustration of concepts. In real study scenarios, we need to consider a more complex interplay of outcome of interest, various sources of bias, necessary level of evidence, and the expected exposure. It should be noted that the direction and the magnitude of bias affects the accuracy of risk estimate and the minimum sample size. In the presence of bias, larger than expected sample size may be required for a viable study. However, even with an inadequately large sample, risk estimate remains inaccurate, and a signal may still be missed due to the direction of bias. Therefore, enhanced technical capabilities should be considered depending on the impact of bias on study results and inference. In addition, if the expected exposure is very rare, descriptive studies may be the only viable option in this framework for signal detection, and the signal evaluation is likely not feasible. If the expected exposure is not very rare, multiple viable options might be considered. The threshold for very rare may differ by scenario. It would be difficult to precisely estimate the expected exposure. Therefore, the refined categories are desired. One important concept not illustrated in this example is that the viable studies may be used alone or in combination, either sequentially or simultaneously to improve efficiency. The final step of this framework is to select optimal study from the viable options. For signal detection, one important question is how early a signal can be detected. In this example, studies 1, 3, and 5 may be able to detect the signal as early as year 4, depending on where in the range the exposed pregnancies can be captured. Selection of optimal study considers timeliness, resource requirement, and other trade-offs. In this example, all 3 studies can detect a signal in a timely manner. Resource requirement is considered high for study 1, as it need to establish and maintain a large-scale registry. Both studies 3 and 5 use an existing data source. Study 3 conducts chart reviews, so it may require more resources than study 5. Although the perceived resource requirement for study 5 is relatively low, one trade-off is that the method may not be efficient for the pre-specified outcome X. Using the data mining method, it is likely to generate many false signals, which will require additional effort to clean up. So put all this together, study 3 is selected as optimal study in this hypothetical example. So next, I will walk through an example to summarize the thinking. For drug A, animal data suggested some risk for outcome X. There is no safety concern for any other outcomes from any other sources. With that, the regulatory question at the time of approval is if there is an increased risk of outcome X associated with drug A during pregnancy. Given the limited information, the study goal is signal detection, if detected, followed by a signal evaluation within 10 years of approval. A study using database Z is being considered. However, this study may be subject to non-differential outcome misclassification with 80% sensitivity and a 90% specificity of outcome ascertainment, according to a prior validation study. Based on our assessment, the direction and the magnitude of bias will not change the interpretation of results, so the potential bias is acceptable for signal detection. For signal evaluation to quantify the risk, the study's technical capabilities need to be improved to account for bias. Based on biological plausibility and the clinical judgment, we assume a relative risk of 3 for the drug outcome pair, with an estimated 3% prevalence for the outcome X and the 80% designer power. 2,308 exposed pregnancies are required for signal detection with a 20% type 1 error. In the meantime, drug A is indicated for a common condition that affects women of childbearing age. It's the 5th in class, and there's no pregnancy-related warning or contraindication labeling, or REMS. Based on these factors and the utilization data of similar drugs on the market, the pregnancy exposure is estimated between 1,000 and 10,000 per year in the US population. Database Z covers 40% of the US population, so it anticipates capturing 40% of total pregnancy exposures in the US. Integrating the study's internal validity, minimum sample size, and expected exposure, signal detection is anticipated in year 4. With improved technical capabilities for more accuracy, signal evaluation to quantify the risk may be achieved in year 5. With that, the study is considered a viable option. Comparing with other viable study options, this study can detect and evaluate the signal for outcome X with the required level of evidence in a timely manner. The study can be efficiently operationalized using the existing resources. So this is selected as an optimal study. So this summarizes the FDA's current thinking. Further development is needed to identify potential gaps in the preliminary thinking of the framework, to determine how to estimate the magnitude of drug exposure, especially at the time of approval, and the fraction that a potential study can capture. Also, we need to evaluate how these studies may perform similarly or differently in different scenarios. We hope to learn more from the demonstration projects, which will be discussed in session 5 tomorrow. Based on the additional knowledge, the framework will be refined by the end of PDUFA 7. So finally, I would like to acknowledge our colleagues from various offices in FDA CDER and CBER who made invaluable contributions to the development of this framework. All the information presented here is a product of team effort. Thank you.
All right, Wei, I think you get the opportunity to sit down because that's been a long time through all of that. And I think you probably saw people out in the audience going, how do I interpret all that's in your brain
Yeah.
that you were just conveying to us? So, so I'm going to, I think, kick off some of the discussion, and I'm going to start with our with a few questions directed to our panelists, and give Way a moment to catch her breath as well. So, Leyla, I might ask you, since you've been put on the spot a few times already today. So, that's a lot, and I think it's a lot that everyone's going to need to sort of think through and absorb. But how do you think this framework is really going to then fit into our overall safety assessment for products as we go into this post-approval setting?
All right, thank you for that question, Geeta. So we've all thought about these issues. Like Wei said, it's been a real team effort across different offices and divisions, and so this is kind of our first attempt to put this out there to you, our stakeholders, to get input from all of you, and so I guess. In terms of where it all fits, the framework, I think, is kind of one piece of the overall puzzle. So the overall puzzle is, right, like drugs are approved, they get onto the market, and then we have to figure out how to get safety. And so this is kind of one piece of the puzzle in terms of, okay, trying to figure out what are the ideal study methods or designs. I think there's a few other considerations that we also have to keep in mind in the overall safety assessment of newly approved products. And I think one thing that I wanted to mention that I think hasn't been mentioned yet at this meeting is that industry has access to safety databases where anybody, any patient, consumer, anybody can report a pregnancy that has been exposed to their product. And so we review these annual reports, and what's interesting to see is that we often see significantly more cases reported to sponsor safety databases, disproportionate in terms of, then you look at the numbers of pregnant individuals who are being enrolled into a pregnancy registry, or that, you look at a database, a claims database, or we look at Sentinel, and we just aren't seeing the exposures there. So I think we need to think about how to leverage that data. So although safety, these adverse event reporting systems are set up for the public to report adverse events, pregnancy is kind of a different kind of situation, because folks are willing to report pregnancies that are occurring, and so this is an ideal opportunity to collect data. So, we need to figure out a way to then get good follow-up data on those pregnancies that are being reported, because they're not necessarily just adverse events, right? They're actual exposures of pregnancies that are occurring, and often they are reported during the pregnancy. And so this is a great opportunity to do really good prospective follow-up and get those outcome data in those patients, and try to see what we can do with that data.
Sara, do you want to add to that?
Yeah. If I could add one thing, because I think it's been mentioned today, and we've talked a lot about this, that we recognize that the individual patient benefit risk has to take into account many factors: what the patient needs, what the benefit of the drug would be, and importantly, what other treatment options are there, and importantly, what is the risk known about this drug. And so we just want to be very clear, as we've talked, that this is to fulfill that safety piece of the puzzle. So just want to put a nod out to the other comments that have been made about the importance of the overall patient benefit risk.
So picking up on that then, do you see this framework then helping to sort of provide a way to address some of that gap at the patient and provider level on what kind of information might be helpful or be known?
I'll let others comment. As with the same goals as we've talked about today, as all of the safety studies are intending to do, but I'll let my colleagues add anything to that.
Yeah, just to add to that, I think, as Sara said, thinking about the benefit of risk, so we need data for risk, talking about the risk. And this framework really helps us to identify the optimal use of the studies to generate data for risk. So we can consider in the ecosystem how to use those information.
So there's a lot of, I think, kind of assumptions that are going into this framework, right, that are sort of the ways to develop these strategies, the sort of study designs and so forth. So one of the things that is really important is about the numbers, right, meaning, well, there's a lot of numbers, but the numbers of expected, right, sort of expected cases, expected exposures, actual rates. You know, how do you think that those things are going to come into play when a sponsor or a target group, you know, sees a condition as being really important, but maybe it's a rare condition, ones where they're very common, but people don't really see this as a significant concern. So there's going to be these sort of things that come into play when trying to decide what's the optimal design, even though that might not be what the going in thought might be. So how do you think that's going to
I don't know if
address?
anyone wants to come to first. I can go first. I think that's why this is so complicated.
Yeah.
And that's why we needed a framework to help us think this in a more structured way, using a consistent approach. So there are so many different numbers, different factors we need to consider at the same time. So we don't want to really focus on one aspect and forget the other one. So we want to put everything together on a list so we can think of everything at the same time to evaluate their importance in different scenarios. So I think it's really about how to put different thoughts, different important factors together.
So I know there's going to be lots of discussion on this tomorrow as well, but it strikes me as the starting off point is there's approval of a drug or biologic or product, and then it isn't just go do a post-market evaluation. It's going to be discussion about what's maybe the best approach to doing the post-market evaluation. What is the right strategy to take? How can that be something that is discussed together? Is that conceptually what you all are thinking?
Right, yeah, I think as Leyla mentioned in the morning, there are different approaches. Not only the non-interventional studies, we also have pharmacovigilance. So the data come from all different approaches. But for non-interventional studies, because there are so many complicated factors, we just need to have this framework to help us to sort out the issues. I think that's a
Question?
Other questions for Clara?
Okay, sorry, yes, I'm sitting back
So
there.
just one fundamental question that I think might be easy to answer, and that is, in your example, you landed on one study type for a drug. Is that the goal, that you're going to land on one study type for this goal for a drug, or that you're going to end up with a sort of a group of studies because of all the different biases in different directions and so on?
Anyone want to comment first?
You can answer that
Oh,
one.
okay, I can go first. Yeah, it depends. So yeah, I think the framework has enough flexibility for us to deal with different scenarios. And then we do think about what is the best way when we consider all different factors at the same time. If it's more efficient to have multiple studies simultaneous or sequentially, we can go that way. Or if one study is adequate, then we don't have to use additional studies.
When do you envision this coming into the planning, or the discussion? Meaning the decision about the right approach or the discussion with sponsors? Has anyone thought through that yet?
Well, post-marketing requirements are issued at the time of new drug approval. So ideally, these types of discussions should be happening prior to those decisions, prior to drug approval.
Okay.
Yeah.
Okay, that was said ideally, right? So we'll see what
Yeah.
ends up. Question over there.
Hi, Evan Myers from Duke. A lot of these considerations are going to be the same for a given condition or class of drugs. And would you envision every time a new drug is approved for a given indication, that the individual sponsor would have to go through this again, or could there be kind of a library of considerations for a given class of drugs for rheumatic conditions or hypertension that could then be shared, so that, again, to improve consistency and comparability and all the things that we ultimately want?
Anyone want to ask?
Yeah, I can.
Okay.
And then I'll turn it to you, Wei.
Okay, yeah, go ahead.
So if there are parts of the framework that are common, I would think they can be replicated, reproduced. But the studies, for a given study designs technical capabilities, that's, I think, where the biggest difference, the biggest customizations happening, or the biggest, difference that would have to be, really thought through. I don't know, Wei.
Yeah, I agree. Yeah, I completely agree. Yeah, I think the framework, as we designed, it's not for a particular therapeutic area. It has the flexibility. So if additional factors should be really considered, we can definitely add that to the framework. So I think a more thorough thoughts can maybe after the workshop, after we hear back from the stakeholders, we can think better how to refine the framework.
Jeff Roberts from Merck. First, I just want to applaud this work that was done. Very comprehensive set of data on all the studies we have in all these different contexts. And I guess I want to take it to the next step and say, have you started to think about whether we reached an optimal endpoint for each of these products in terms of the labeling that we got to for these studies that were done? And I guess maybe to take that even a step further, have you thought about taking this new approach to this framework and applying it retroactively to some of the studies that have been done, and saying, would this have worked better or different if we had this framework back when we were thinking about it then?
Leyla, do you want to answer that?
All right, you're asking us to do a lot of work, Jeff. I think we have enough on our plate with these commitments, with all these demonstration projects that we're going to have to complete by the end of fiscal year 2027. So I don't think we've thought about doing any kind of retroactive type assessment.
Well, I might pick up on Jeff's question though and say, let's say there's something that the label has been changed based on some data that's out there, right? It might be a question to say, was that what the approach that you would have chosen if you'd gone through this framework? Just sort of like as a hypothetical, right? To say, take, right, right.
Enough,
Yeah, because I do think that I'm looking at this from the standpoint of a research investigator, and it's almost sort of like when we go back to clinical trial design and go through the phases and understand, right? This is going to take people a while to figure out how to get the information and the inputs and understand what they even mean, because you're talking about a lot of, the data's there, but how we put it into the terminology that you're using is going to take some learning, right?
Yeah.
So I do think sometimes using examples, real examples where you can, I think your examples are helpful, but I think it will be even more impactful if you take it in a framework of this drug, this is where it went, here was the pathway, here's what it would have been if we were doing it through this model now. And that might be a way for people to grab that and then understand.
Okay, that's a good idea. Yeah.
Sorry, I turned this way, so I know I'm biased. It's the side of the room.
Hello.
Oh, yeah, oh, sorry. I didn't mean to turn
No
away.
worries. Hi, I'm Elsie Grace from Eli Lilly. I was just thinking about the framework, and thank you. It was a great presentation. I'm wondering how the Sentinel could be kind of incorporated into the framework, or the idea of the sufficiency in the PMRs. So I don't know if there was any thought about that, particularly if some of this discussion is ideally supposed to be happening prior to approval.
I can address that at a very high level. At this time, we're only considering the science. So in the ecosystem, how the stakeholders can work together to generate the best data for decision making. So what stakeholder will do what, that will be the next step.
I have a few questions out here. I think we
Hi. Just a quick question on how do you envision the framework translating into labeling? So Leyla, you had mentioned, spontaneous reporting. Is that sufficient to put in a label?
Well, everything, of course, comes down to the quality and the quantity of the data. So, I thought there was somebody in the back who was making a comment. Oh, okay, sorry.
Okay, we'll come 1 second.
So I think it's an assessment of what data you have, so what's available, and looking at the quality and the quantity of the data. So we all understand the known limitations of pharmacovigilance data, and sometimes that data can be messy, but sometimes that data can also be really informative, and I think we've had several examples mentioned today of how pharmacovigilance data has been useful, and I can also think of another example of a cancer drug, trastuzumab, which is approved to treat breast cancer, and clinicians who were treating pregnant individuals with breast cancer were observing that the amniotic fluid was going down, so resulting in a condition called oligohydramnios, which was resulting in fetal death. That was, these were interesting observations, and then to add on to that, treatment was withdrawn, and that resulted in the amniotic fluid reaccumulating. So kind of like a challenge, de-challenge kind of situation. And then they restarted the drug, and then the amniotic fluid went down again. So, these are based on pharmacovigilance, right? It's like case reports that identified a signal, but the signal was so strong, and it was actually, with this challenge and de-challenge situation, it was actually confirming the signal as well. So I think you have to take every data source on its own and evaluate it for its own value.
Yep, right there in one of them.
Okay, great, thanks. So my question, it seems like from what I've seen just here shortly on the framework, a lot of it is dependent on kind of the sample size estimates and the magnitude of the expected drug exposure, which we've heard, earlier today that sometimes, it's hard to predict, and sometimes things don't pan out where we think they should, and even with some of the larger database studies, sometimes they still have small numbers. So how will that, I guess, allow for some flexibility kind of in real world situations if you go down the path of a particular study design and see that it's not really giving results or numbers of exposures that we expected it to in order to still come to a successful outcome?
Yeah, I'll take that one. I think that's very true that for us to get like a pregnancy study that's successful, the actual numbers have to come out similarly to what we used in the framework or what we projected. I think one thing to think about is like how, I think someone mentioned it this morning about doing feasibility assessments. I think it's, we can't predict the future. It's really important for the protocol to have plans to monitor exposure and capture rates, and also have plans for feasibility assessment periodically. I think Dr. Hua mentioned in the slides mentioned better prediction of exposure at the time of approval. I think that's something we should work on, and hopefully with that, through this whole process, that we'll be able to better predict exposure. And the framework is now incorporating misclassification bias in the sample size requirements. So hopefully with that, we'll be able to kind of minimize the discrepancies, and hopefully feasibility assessments will, after initiation of the study, will help us get more successful pregnancy studies.
Okay, this will be the last question over here.
Yep.
I'm Dinchi Penna from AstraZeneca. My question is for Wei. When you were presenting, you said something about 40% coverage, like 40% national coverage translates into 40% pregnancy coverage. Isn't that a stretch to assume that if a data source covers 40% of the U.S. population, we'd expect to see 40% of the pregnancies, especially considering mother-infant linkage and considering case reviews, right? Because one of your slides assumes that case reviews, chart reviews would give the same number of counts as a database study, which I find that hard to follow. So maybe I'm missing your point.
There's no point there. Hypothetical number.
Hypothetical.
Yeah, it's a
Hypothetical.
hypothetical.
It's exemplary. to
Yeah,
just
yeah.
show
Sorry
how
for the
it works.
confusion. Yeah.
Yeah, right. So, okay. So I think we'll have to close the discussion at this moment, but I just want to thank the panel for their contributions, and I know we're going to have lots more discussion tomorrow, so you all better be ready with the other panelists and so forth. So I just want to remind folks that you can continue to send questions in, and also that there's discussion via the docket, right, that is open and tied to the Federal Register notice. So we'll turn over to Mary Ann Hamilton Lopez to close us off, please. So, okay.
All right, thank you again to all of our participants who contributed today. We heard a lot of exciting developments. Just a brief recap. So we started off the day with the FDA's opening presentation, where the lack of pregnancy safety data in pregnant individuals was emphasized as a public health issue, and that all stakeholders are needed to address this. In session one, we heard from a variety of stakeholders, including those in industry, healthcare providers, and a patient representative. We learned that oftentimes both providers and their pregnant patients must balance the risks and benefits of managing their disease and the safety of the pregnancy, which is a direct result of limited information. With limited data, many patients feel torn between continuing medication while also ensuring the safety of their pregnancy in the face of limited data on medication. Creative solutions for collecting quality data are necessary to improve data availability and accuracy. In session two, we heard that from those who design and conduct pregnancy safety registries, the challenges and advantages of each type of registries were discussed. The antiretroviral pregnancy registry was highlighted in its success, but it required a wide range of expertise and a firm commitment from various stakeholders. Another key lesson in the success we learned from the CDC's COVID-19 registry is how critical it is to clearly define outcomes of interest, ideally narrow in scope rather than open-ended, as well as plan specific analyses prior to implementation to guide the data collection. Then in session three, we heard from FDA staff about recent work in the post-approval pregnancy safety space. Their presentations concluded that pregnancy safety studies have not had sufficient time to mature and inform the regulatory action, with only 46% of the studies informing labeling updates. Next steps for studies examining product utilization data include further exploring years of approval and disease and using these updates to inform the development of the framework. To optimize post-approval pregnancy safety studies, we covered the importance of considering the outcomes of interest, study goals, studies' technical capabilities, and magnitude of drug exposure. Building on these learnings, Session 4 covered how the pregnancy safety study framework operationalized these important considerations into a consistent and transparent approach and how we may be able to optimally obtain timely evidence of safety for regulatory decision-making. As we conclude today, I'd like to remind and encourage, again, this audience to contribute to the discussion via the docket. It's currently open, and the docket is seeking broader feedback from the community on this very framework that we're talking about. You can find the link to the docket via the Federal Registry notice for this public workshop, and it will be open to November 30. We're just going to keep saying it. And with that, we will conclude day one of the workshop. We look forward to reconvening tomorrow at 10 a.m. virtually and in person here at the National Press Club. I've been asked to remind you, if you came in person, please bring back your badge. And we will be having tomorrow a live open public comment where individuals who have previously requested to provide an oral comment on post-approval pregnancy safety studies will have the opportunity to do so. If you are interested in participating in this open public comment period, please reach out to our staff at Duke Margolis. After the open public comment session, session 5, we'll discuss existing gaps in the design and performance of different pregnancy safety study types to better inform the development of the framework. We will learn about the expected demonstration projects that aim to inform future development of the framework that was presented today. The last session tomorrow, session 6, will have targeted discussion with select stakeholders in the community to provide additional feedback on the FDA's pregnancy safety framework. Again, thank you to all the speakers and panelists who shared their work and insights with us here today. We covered a lot of ground. And then finally, thank you, everyone who joined us virtually and in person. We look forward to seeing everyone tomorrow morning, and have a great afternoon.